Thanks again for inviting me. I'm here to present our paper on multi-domain distribution learning for de novo drug design. And I hope that at the end of the talk, all these different parts of the title make more sense. The structure is roughly that I want to motivate the problem first. So why do we care about de novo drug design in general? I guess that part is particularly interesting for the people who come more from a machine learning background and maybe are not so familiar with the kind of challenges in the field of drug design. And then I want to touch on the different technical advances of the paper, the kinds of benchmarks we ran, and just like present the results in like different chunks. And then towards the end, if there's enough time, I also have some outlook, some material that is not in the paper, but what we're currently working on and where we think this line of research is going in the future. So I hope I get to that too. But let's dive right in. So basically, this first slide is really to catch all of you, I hope, because I'm sure that you've seen this kind of law. It's called Moore's Law. Gordon Moore was one of the founders of Intel, the Intel Corporation, and he basically predicted that the number of transistors is roughly doubling every 2 years, which means there's an exponential growth in compute capabilities. And this is, of course, something we're leveraging every day now with our machine learning research and all kinds of computational research, basically. But this stands in stark contrast to what drug discovery is experiencing, because there's this kind of inverse Moore's law, which is also why it's sometimes called Eroom's law, which is really just more inverted. That states that the efficiency of drug discovery is actually declining exponentially. And we're now at a point where developing a single drug costs more than $1 billion US. And this trend is continuing, it seems. So there's some clear issue here. We want to get new medicines. We want to treat diseases that are currently not treatable, but it seems to get increasingly expensive. But the nice, let's say, coincidence or pattern we observe here is that we have two exponential trends that are kind of inversing. So the hope is that with the increasing computational capabilities, we can kind of make up for at least some parts of this decline in drug discovery efficiency. And now I want to get into why the efficiency of drug discovery is declining, but I'm going to make it very, very brief because drug discovery is a very long process that has many different aspects. And I guess all of these aspects can be improved and streamlined to get drugs to market faster and more cheap. But I want to focus here on the very early stages, hit discovery. And basically, this is the task of, you already found a protein, let's say, that is involved in some cellular process related to a disease of interest. And now you want to drug this protein. So you want to come up with a small molecule that binds to the protein in a specific way to trigger some effect. So this is what we're talking about. And then after that, there's some downstream optimization of that original molecule that you found, and then clinical studies and so on. But just focusing on this hit discovery, how people usually do it right now is typically some form of screening approach. So you have your protein of interest and you have some compounds already available, could be in just your, you have them on stock in your company. And then you run them all in some high throughput assay and check if one or several of them have some initial effect, measurable effect. So you really start with like hundreds of thousands to millions of compounds, and it's a brute force approach where you try to find one that has some effect. And then you might find one or several, and they trickle down this drug discovery pipeline all the way through the clinical phases. And at the end, you might have one drug if you get lucky. But actually, the overall success rate of a drug discovery program starting and making it to an approved drug is overall still very low in general. So one striking fact is that the number of drug-like molecules is actually much, much bigger, estimated to be much, much bigger than what we're currently screening physically. So I told you that some libraries go up to millions of molecules that can be screened, but the estimations for how big the chemical space is that is interesting for drug discovery applications go up to 10 to the 60. Of course, this is already like a large estimate, but let's say even the most conservative estimates are not lower than 10 to the 20, 10 to the 30. So it's like really orders of magnitude different spaces that we can search. But of course, with physical screens, we just hit limits. We can't possibly ever search this whole space. We would have to physically have all these compounds. And here, this is where de novo drug design, especially computational de novo drug design, can play a role and make a difference in our opinion, because it allows you to make on-demand molecules that are more customizable. You have more options to optimize them already in the computer before you test a single thing in the wet lab. Very like common instances of this approach are structure-based drug design, which is what I'm focusing on here because this is what this paper has been about. But there's also ligand-based drug design where you don't necessarily know the protein structure that you're targeting, but you have, let's say, some other molecules that you know are binding to the same binding pocket and you can extract patterns from there. But again, here focusing on structure-based drug design. And just to give you a more concrete example where this can be particularly useful is a drug category called induced proximity drugs. You might have potentially heard molecular glues or protacs. These are all from this like same family of drugs, where the idea is that if you have some protein target of interest that has a very shallow pocket where it's hard to design or find a molecule that binds to that pocket, what you might be able to do instead, even though you might not find this small molecule that binds directly to it, is to recruit a second protein that also has a flat interface, and that then triggers the effect. And this recruitment is basically it's mediated by the small molecule drug that you're designing. So this small molecule glues them together. That's also why they're called molecular glues. And then this can trigger some downstream effect. And the defining feature of this kind of therapeutic class is that you have a lot of geometric and chemical constraints for your small molecule, because now you're modeling a complex that has proteins on both sides. So you have like, steric constraints on both sides, but also, let's say, hydrogen bond donors, acceptors on both sides that need to be satisfied. So there are a lot of constraints. And the more constraints you have, the less likely it is to find any fitting molecule that fulfills all these constraints in your physical screening library that I told you about before, which is kind of the de facto standard approach right now. So here, the de novo custom design really can make a difference for these kinds of drugs. And before I get into the contents of the paper itself, I also want to give you a rough overview of how I see the drug design field for now. Of course, it's just I split it into two big categories just to set the stage. Of course, there are many more subcategories. But as I see it, the first big category is some kind of optimization-based approach. And here I'm thinking of reinforcement learning, for example, where you have some reward function and your agent learns to really improve that reward function as much as it can. Or search-based approaches. There, for example, some Markov chain tree search approaches, or some people have trained variational autoencoders and search in their latent space to find molecules that optimize a specific objective or reward function. And this kind of class of models has very unique advantages and disadvantages. So obviously, if you really optimize for one particular target property, you will get molecules that have really nice properties according to in this one axis. You also have a lot of control over your action space. So for example, what people are doing already now is that that each, let's say, branch in this tree, for example, corresponds to some step in a chemical synthesis. So that at the end, let's say your first node is a building block, and then if you branch out, you add another building block with a specific reaction. So at the end, when you design a molecule, you actually have some kind of synthesis recipe, and you already know how to make this molecule in the lab for testing. Additionally, you don't need a lot of training data because, a lot of this stuff, for example, reinforcement learning is built also on simulation and having good computational oracles. So you don't need to a priori have that data, as long as you have enough compute to simulate and gather enough rewards to train the model. The downsides are that it's rather inefficient because you get this very little signal. Let's say after a long trajectory, you get one reward out, and based on that, you need to optimize all your parameters. It's also hard to generalize to more complex design spaces. So for example, this 3D structure-based drug design that I showed on the previous slide is not easy to tackle with these approaches. And actually many people are not currently doing it for that reason, because the whole setup just becomes much more complex very quickly. And some of these approaches also suffer from mode collapse. So basically, once you find one good solution, you just stay there in this local optimum and never explore. So then we have the second class, which is kind of complementary in its advantages and disadvantages, in my opinion. And I call it distribution learning. And what I mean by that is, for example, chemical language models, which take like a SMILE string that represents a molecule, for example, and like a huge collection of smart strings, and it learns to generate other smart strings that look like those. So it learns to generate molecules that look like the ones in the training set. Similarly, the fusion models in 3D do the same thing, just in 3D, also generating the coordinates of the molecules at the same time. And here, yeah, as I said, the advantages and disadvantages are kind of complementary. So the objectives are now implicitly encoded in the training data, which can be an advantage because it means you don't necessarily need to be able to quantify exactly what you're optimizing for. For example, to give you a concrete example, if you just want something that is biologically active, you might have a data set that you measured already and you know they're biologically active, but you can't really say why, and you can't define a computational score that tells you that they're active. You just know they are because you measured them in the lab. So this is one example for this implicitly encoded training objective. Then you also learn very abstract representation of this whole design space if your data set is big enough, because you kind of, like a large language model, you really learn some like foundational information about the data in these kinds of models. You also typically have more diverse outputs because you're not really optimizing for one particular mode in your distribution, but you try to cover all the modes if you do it properly. You're not suffering from any problems you might have with your computational oracles because there is no computational oracle. You just need to make sure that your data set is clean. And yeah, it's easier to expand to 3D cases, for example, the structure-based struct design that I'm showing here on the slide as well. But of course, you might see like language models, especially without fine-tuning, they still seem to hallucinate a lot and make up facts or things. And this happens with chemical generative models as well. Plus, if you're too free in what you're designing, you also always suffer from the synthesizability aspect. So you might design some molecule, but you don't know how to make it. And basically, no one knows how to make it because it's too dissimilar from anything that has been synthesized before. And I already alluded to the fact that to train these models properly, you need a lot of high quality data because only then they actually learn something meaningful. But I'm ending on this category because this is actually also the category that the paper we're discussing today fits in. So it's about distribution learning. And so this actually also sets the stage for the reason we worked on this, or like the main premise of the paper, which is we need to do the distribution learning properly. And on this slide, I want to motivate what I mean by that. So typically, the loss of a distribution learning model, a generative model, looks something like this. Here, I just copied the diffusion model loss from a paper. And what you're doing is you're optimizing the likelihood of all your data points in your data set in some form. So the details of the loss function don't really matter here, but it's important to note that you're really optimizing the likelihood of data points under your model when you're training a diffusion model. And then if you do this in the molecule generative space, you might see a lot of tables like this for evaluation. This is actually taken from my very first preprint of my PhD. But also to show you, I'm not the only one who's evaluating like that. I'm showing a second example from a related paper. They all look something like this. And you have a bunch of metrics that are more or less relevant for drug discovery applications. Just to name a few, you have this VINA score, which is a proxy for binding affinity, how strong the molecule binds. You have QED, which is like a heuristic for drug likeness. So basically some patterns that you wouldn't find in a drug would score low in this QED score, while if your molecule looks like drugs that have been discovered before, it would score higher. SA is synthetic accessibility, so it tries to predict how easy it is to make a molecule, and other scores like that. And then typically people bold the numbers in the table whenever they achieve the best scores in these metrics and say, hooray, we have the state-of-the-art model, and then they can publish a paper. But then, since this has been quite successful for some time, something interesting happened at some point where if you look at these numbers, for example, the models got better and better at all of these metrics. And then this particular model here, for example, scores better on this binding affinity metric than the reference molecules from the training set. Or in this case, it's actually the test set, but let's say they're taken from the same data source. And this is, of course, a bit hard to explain. If you're just optimizing for the likelihood of your data points, how can you score better? And what I think is happening here is that basically people over-optimize the hyperparameters to score well on these absolute numbers of the metrics, while it's actually not really what the model itself is optimizing for. What I mean by that is if you just kept training longer, you would probably not improve on these metrics anymore, but actually get a bit worse again because you get closer to the training data distribution, if that makes sense. So this leads to the approach that we've been pursuing in the paper for training generative models for drug discovery. And yeah, I just basically list the recipe here. Usually you start by collecting as diverse and high quality training data as you possibly can to train your model. I want to mention that this is out of the scope of this particular paper because for benchmarking purposes, we still used like a very commonly used data set for that people used in the past, so we didn't have to retrain all the baseline models. But in principle, this should always be your first step. Get the best data set possible. Then you train your generative model, and then you benchmark it, but not looking at absolute numbers, but really focusing on the distribution learning capabilities. How well do the molecules you generate match the molecules in different aspects of your training set? And then there's a fourth step, and to motivate that, I want to show an example. It's maybe already a bit outdated because now these AI image generators are actually quite a lot better. But there's still a lot of hallucination going on if you learn distributions with a generative model. And this might be fine if it's just a fun like image that you're generating. But if you're generating a potential drug that you want to get to the market and that you want people to take, you really need to make sure that you're getting rid of these hallucinations and yet you're still optimizing for certain properties of your molecule and not just take them anywhere from the distribution. Because typically the good molecules you want, they're not necessarily near the main modes of the distribution, but maybe somewhere in the tails. So you need to extract relevant samples before you continue. And here, different versions are possible. I'm mentioning just two on this slide. The first one is fine-tuning of any form, where you just shift the whole learned distribution to something that you prefer. And this is actually what we did in the paper itself, and I'm going to get there later on. But then you can also do certain sampling tricks and optimize later on at inference time. And if we will have time for the outlook, I will also say a few things about that approach. Okay, but with that, we can finally get to the actual like technical aspects of the paper. We call our new molecule generative model DRUGFLOW, or there's also an extension, I'm going to explain it in a second, that we call FlexFlow because it basically has flexible side chains in addition to generating the molecule itself. So I just mentioned some of the core features, especially the ones that are a bit different to what has been previously done in prior work. So for example, we're learning now on we're learning data on living on many different domains. So we have the coordinates, which just live in Euclidean space, the atom coordinates, and these we can model with a flow matching based approach. Then we have the atom types and bond types, which are categorical variables, so they live in some discrete space. And here we are applying some form of discrete diffusion to generate them. And then finally, this FlexFlow extension also has sidechain modeling. By that, we mean we're generating the small molecule, and at the same time, we're also estimating the bound conformation of the protein pocket itself. But there are a lot of constraints on these atoms in the pocket, because basically you have a few rotatable angles that you can change, but they determine the whole 3D structure of the pocket. So actually what you want to study here is not like a Euclidean space, but some manifold of these angles. And that's why we're showing it with this torus here, because this is actually a hypertorus in the end. And here you can perform Riemannian flow matching to model this. Then this whole part in the middle, this yellow part, is basically just some technical detail of how we represent the data in the computer and how we run the flow matching / discrete diffusion framework. But then we have some more practical features of this model that are also different to previous models. So for example, we do not have to specify the ligand size exactly in this model anymore. Meaning, so basically, most of these models are restricted because you have some computational graph. Each atom is a node in a graph, each bond is an edge, and you need to define the size of that graph before you start computing anything. But that also means your ligand size, the molecule size you're generating, is already predefined. The model can't really do much about it. And here we found a small trick, a workaround to have the model actually decide on the size. And I'm also going to show you why we think that matters. And finally, you are probably familiar with AlphaFold, where it's like this protein folding tool, and it comes with a confidence score. So basically, with each prediction, you have a confidence of the model, and people use this very heavily for all kinds of scoring, ranking, just evaluating how well their predictions work of AlphaFold. So we thought it would be nice to have something like that here as well. And we developed this per atom uncertainty score that comes with every generated sample. But this is just an overview page. So now I'm going to get into the different components one by one. And I really invite you to ask questions basically whenever relevant, because I will just show a bit more technical detail and then some results, and then move on to the next component. So the first thing I wanted to show is how we are evaluating the distribution learning capabilities, as I mentioned. So now the slide will be very busy with just a lot of tables, but the core takeaway is we take some of these metrics that I've showed you before, so the drug likeness scores again here, synthetic accessibility, we're measuring some hydrophobicity. So all these are metrics that people evaluated in the past, but they really look for absolute numbers. What we did in this paper instead is we compute all these metrics on our generated set of samples, and we compute them on the training set, and then we compare the two distributions. And we compute Wasserstein distances or Kullback-Leibler divergences to assess how well we're matching the training distribution. And that's actually what we're reporting here. So lower is always better because it's always distribution learning differences. And yeah, we see in the top row, DRUGFLOW does quite well. It's usually the best model when we compare it to some other popular alternatives. We also have a full table dedicated to all interaction-related things. So for example, we have here these docking scores again, but we also counted hydrogen bond interactions or pi stacking interactions, trying to make sure that we're generating roughly the same number of hydrogen bonds, for example, as training examples. Then another interesting thing is this FCD score. It stands for Frechet-Chemnat distance, and it's very related to Frechet inception distance that you might know from image generation evaluations. So Chemnat is a pre-trained property predictor. And for the FCD score, people took this Chemnat model, feed in some molecules, and then they get the embeddings. Basically, before the prediction is made in the model, they take these embeddings. They fit Gaussians to all the embeddings, and they do this for two different sets of samples, and then compute the Frechet distance between these two Gaussian distributions. And that's the FCD score. So it's a bit of a more abstract way of measuring how similar two distributions are. And also on this one, the gap is actually particularly large between DRUGFLOW and the other models. And just to give you a bit more of a visual idea of what that means is I also included this little picture here where we took these embeddings, mapped them into 2 dimensions so that we can look at them on a slide, and then show in blue the training data embeddings, what kind of space they cover, and then with these like black lines, where the embeddings of the samples fall. And here we can see that most of these models we compare to, they just cover this main mode of the distribution more or less well. But they miss the second mode completely. While DRUGFLOW, well, first it samples some molecules from the second mode as well, and it also just more nicely covers the whole main mode. So this is where this difference in the FCD comes from actually, visually. Okay, so if there are no questions on the general distribution learning, I will move on to how we do the flow matching on the sidechain angles of the protein. The general idea is, again, we have this hypertorus on which these angles live. So if you have 2 angles, the torus will actually look exactly like this. With more angles, it's a bit harder to visualize. And then you do flow matching. And I hope people are roughly familiar with how flow matching works, but basically you're matching a vector field, usually in Euclidean space. In Riemannian flow matching, which we're doing here, you first map data points from your manifold onto the tangent space at that point, which is shown in this blue plane here. So x is the data point, and the blue plane is the tangent space at x. And then you can define a vector on this tangent space, which is like a lower dimensional Euclidean space, well, tangent to the manifold. And you can do everything else in the exact same way, just in this tangent space. You map to a new point, for example, if you're following your flow, and then you map that point back to the manifold. And in practice, what this looks like, I'm showing here with this animation where the side chains move while the small molecule is being generated and denoised. There's a small trick that we empirically found to work better than, let's say, the standard approach. So for that, you need to know that in flow matching, people usually have a linear schedule where noise is just removed really according to, like a linear scheduling function. But if we do this both for the coordinates and the sidechains, we found that this introduces some clashes. So what we found useful was a different schedule that removes the noise for the sidechains a little bit faster, following this blue line here, instead of the gray. So that basically, you could also see that in the animation. Maybe I'll play it again. That the sidechains kind of stop moving while the coordinates of the small molecule are still evolving. And that's exactly because of this different schedule. And this just helps the model empirically to condition better on the sidechains that it generates at the same time. Again, we looked into distribution learning capabilities. So what I'm showing here is the first two sidechain angles. Some amino acids have more than two sidechain angles, but it's just hard to show them on a slide. That's why I'm limiting myself to these two. And I'm showing the reference, which is the training set again, and some samples in the blue lines. And you can see that, again, we're covering quite nicely the different modes.
Right. I have some kind of question on the previous slide. Yes, about flow matching. I was expecting, like, I think you talked about it, the concept of velocity field.
Yeah.
Yes. So is it the one that is influencing the sort of like the ligand atoms within sort of like the ligand atoms within the field in a way they are binding?
That's the gray schedule, basically. Yes. So the velocity field effect, there's two different velocity fields. One on the coordinates of the ligand that affects how they're binding, where they're placed at the end of the process. And then there's the second velocity field defined on this tangent space here, which defines where the side chains will end up.
Okay, makes sense. Thank you.
And this difference really just means that the side chains, the noise, so this kappa t is kind of proportional to the amount of noise that is still in the sample. And this drops more quickly for the side chains, which means they get denoised faster and then don't move much anymore. And then the model can condition a little bit more on what it already sees to generate small molecule positions of the atoms.
So how does the velocity field influence the evolution of these small molecules, but also the sort of like the side chains? Yes.
So the velocity field is really what governs it. Maybe I should have put also one more slide on flow matching itself, because it's kind of the basic principle is that you're learning a velocity field and then you're simulating like a particle in this velocity field when you're generating a new sample. So the velocity field itself is learned, but during inference, you really just do like a Euler integration scheme to get to a final sample following this field.
All right, sure.
All right, then I'll move on. So yes, we showed that the distributions match quite well the training distribution of the sidechain angles, which again just underscores how that DRUGFLOW is like a good distribution learning model. Another interesting experiment we did was, can we recover bound configurations of the sidechains? And here we applied like a little hack. So basically, while I showed you the model generates the small molecule together with the sidechain conformations usually, we can kind of force it to generate the same molecule by just like overwriting what the model predicts. Let's say the model wants to move an atom in a certain way, we just overwrite it with the velocity field that we know will give us the ground truth molecule. And then we just let the model freely decide on the sidechain rotamers. And the idea here is that we force the small molecule in a certain position, and then we want to see if DRUGFLOW or FlexFlow in this case is capable of recovering the bound pocket configuration that fits this small molecule. Yes.
Which atoms? Which atoms are used for this RMSD?
That's the atoms of the side chains here. These rings in gray that you see.
Okay.
All these sticks in gray.
Oh, sure.
And this one is fixed. This is what I just meant with we hack into it and make sure that they are actually end up in the same spot as they are right now at the end of the process. But the model can still decide on where it wants to put all these side chains here in gray. And then we measure the RMSD, which is really just like a distance, an average distance basically, between the ground truth position of the side chains and what the model generated. And then we start from an unbound position, so basically removing the small molecule and just like physically relaxing the side chains, what they would be if there was no small molecule. That's the distribution shown in red. And then what I just described before is the distribution in blue, if you then let the model generate again a bound position. And even though the shift is not huge, it's still notable, like it goes down. And also for reference, this one is the prior distribution. So this is what the model actually sees. So the model never sees these like unbound positions. It starts from here, right? From something much higher and manages to denoise it such that it more accurately reflects the bound position than this red unbound distribution. Okay, that's already the last piece of data I had on the flexible side chains. So if there are no questions, I will move on to how we make the model decide on the molecule size. And here the trick is rather simple and more or less empirical, not very theoretically grounded, I would say. So basically, in addition to this molecular graph that defines the molecule itself, where every node corresponds to an atom, we are also introducing a new so-called atom type, like it's a virtual atom type. So we just label them as no atom or as virtual or dummy or whatever you want. And we just add these to the molecular graph for generation during training. Sorry. And then because we don't want to bias the model too much so that we give it like a huge hint of how many atoms should always be virtual, we're not adding the same number of virtual nodes every time, but we sample this number between 0 and 10 randomly. Like sometimes it gets 5 virtual atoms, sometimes 1, sometimes 10. And then one thing that needs to be considered when you add these virtual atoms is they also need to have a coordinate assigned to them. And that's of course not trivial. There's not one right solution here. But what we found to work is to just assign the center of mass to all of them and train it like that. So basically then you do your noising process the same way for these virtual nodes as you do it for the atoms. And the model learns to approximate this vector field that would push all this back to the data manifold if you run this process in reverse. And to evaluate if this is useful, we came up with this little experiment where we took one protein pocket where I think the original molecule, the known ground truth molecule that binds to that pocket has 17 atoms, I think.
And
yeah.
It looks like the previous slides, you're adding the virtual nodes at the center of the sort of like the ligand mass.
Yes, exactly.
And does that have like, it is intentional, like adding it anywhere would create a different result, or like adding them at the center is intentional, it has specific sort of like interest?
It just needs to be a predictable choice, right? It cannot be too arbitrary. Let's say sometimes you put them in the center, sometimes 10 angstroms away from the center, because then the model doesn't have like a target to regress towards. We found this to work, but to be honest, it was also the first thing we tried and we didn't explore this much more. I know that there's some work, I think, if I'm not mistaken, where they did something similar later on. But instead of putting it in the center, they had like a little Gaussian blob. So they sampled also positions around the center. That also seems to work. But for us, it was fine to just keep it simple and put them all in the center.
Okay, I was thinking maybe at the center, that way you prevent some sort of like mislabeling by the model, because now it knows where the virtual nodes are. Maybe it can label action nodes as virtual. But yeah, thank you.
Yeah, I guess that plays a role as well. Coming back to the experiment, so how basically it's not clear how you would evaluate that this is working well or not, right? So what we came up with is this task where we take one protein that has a known molecule bound to it that has, if I remember correctly, 17 atoms, the ground truth one. So we start from there and we generate a bunch of new molecules that all have 17 atoms. But then we gradually increase this number of atoms. So we ask the model, give me one with 25 atoms, for example. And if we have no virtual nodes, that's the red line here in the plot, So it's kind of the same model trained again without this virtual nodes feature. Then you get more and more clashes as you increase the size, just because you run out of space in this binding pocket. So you cannot add more atoms because there's not enough room for them. So the model necessarily introduces some clashes. And this trend is almost linear at some point. Because you just add more atoms, all of them will clash. While the model with the virtual nodes actually manages to keep this number at almost zero. It almost doesn't introduce any clashes. It's important to note that this plot is within the bandwidth of the model. Basically, it's the ground truth size plus, yeah, like 10 or even a bit more, what we're showing here. And in that area, it manages to do this. But if we go beyond, this is now shown in this like middle plot, the model doesn't manage anymore. And also with this virtual nodes, it goes up. And that kind of makes sense because it learned the distribution from the training set and it never sees more than 10 virtual nodes. So it's out of distribution further and it doesn't do this. So then it kind of evolves the same way as the model without virtual nodes in this experiment. And just as another piece of evidence that it actually what prevents these clashes is that the model removes the nodes. We're also showing the number of removed atoms in this rightmost plot, where it also kind of converges at around 10 to 12 atoms. So it doesn't remove more than that. And again, that can be explained by the training in which it never has to remove more than 10 during training.
I wanted to ask you about like how can someone formally think about the bandwidth in this context?
That's why we put it in the quotation marks. It's not like an actual like information theoretical bandwidth. It's more like what we mean by that is the bounds of the training setup, basically. What the maximum the bandwidth is the minimum to maximum of the training setup, what we trained it on.
Okay. Sure.
All right. So then I move on to the uncertainty estimation. And here, I guess, important to notice that typical in many cases in biology, let's say for docking models, for example, if you want to evaluate the uncertainty, what you can do is you train your model, and then after you trained your main model, you train a second one on top that tries to predict the errors that the first model makes, right? Because if you dock a molecule, there's actually a true answer to this task, and you can compute an error, and you can just estimate with the second model how good you are in the first place. And this is how a lot of the uncertainty scores are trained. Actually, also the AlphaFold one is trained like that in the end. With a generative model like DRUGFLOW, it's not as straightforward to have an uncertainty estimator, because basically if you give it an empty protein pocket, it might generate a lot of new molecules that look nothing like the known binders that you know for this pocket. So you can't easily define an error and just regress that error. So what we came up with instead is the following. So here I'm showing the flow matching loss. It's just really this matching a vector field, a ground truth vector field with your neural network. And then you have a flow matching model that does this. So it does this regression task. It predicts one value. And now the idea is if you, instead of predicting one value, you predict a whole distribution of values. Trying to estimate into what range can a true answer fall. So you have something like this. And then you want this distribution to be easily parameterized so you can predict it with your model. So we actually just decided to use a Gaussian distribution because it has two parameters only, the mean and the standard deviation. And if you then do this, you can actually, by just estimating the standard deviation on top of the mean, you have already an implicit uncertainty value assigned with every prediction. And this is something that people have done quite a lot in the past for regression tasks, but here we kind of expanded this and used this in a flow matching setup. So mathematically what this looks like is you have your, to derive now basically the loss function, how we can train this additional output head is the following. So again, we start from the likelihood of any data points under the model. And now this is the Gaussian distribution with the notation from the flow matching setup plugged in with the velocity fields. So just writing out this Gaussian looks like this. We can compute the log probability, just taking the log on both sides. And then what we want to optimize with the model is this likelihood, like the likelihood of getting the right answer given this Gaussian model. So we computing the argmax of this with respect to the network parameters, and basically this constant term disappears, and we get this thing. And then taking the negative of all that, because instead of maximizing, usually in machine learning, we think about minimizing objective functions, we arrive at this loss function. And the nice thing is that this part here is actually exactly the standard flow matching loss, so it reappears in this loss function. And then additionally, it has a very nice intuitive meaning, these additional terms that now appear here as well. So basically, whenever the model thinks its prediction might be wrong, and this term is large, in order to make the overall loss still small, it can assign a larger standard deviation, or a variance in this case, sigma squared. And then to make sure that it doesn't just predict a huge, like infinitely big sigma square all the time to bring the loss function down, there's this additional regularization term here. So if it just goes up all the time, it will be punished by this term. So this is the intuitive explanation behind this loss function that we're training the uncertainty estimator with. And then this is now in every step of the flow matching sampling process, we get one of these values out. So to get a final value, we just decided to integrate along one of these sampling trajectories and assign, so that's what's shown here on the top right of the slide. And this is the final uncertainty score that we have on each atom. And then we can look at the samples of the model and really assign an uncertainty score to each atom, where now red means high uncertainty and blue means low uncertainty. And we think this can be useful in practice because, for example, if you generate a molecule like this, the model seems to be pretty confident in this ring system down here, but it has some issues with these additional rings. So for example, as a medicinal chemist, you could now go ahead, remove these rings and like replace them with something else, or let the model regenerate these parts, for example. But you keep this like ring system that the model already liked, for example. I think in the interest of time, I will move a bit quick. I think the most interesting result we had with this uncertainty estimator is the one you see on the bottom of the slide, where we again plotting some very simple metrics. So it's just bond angles and bond length, the most common ones. And we're plotting the distribution of everything that is generated, and we're coloring each bin by the average uncertainty that the model assigns here. And we can see that this uncertainty really tells you quite nicely where the main modes of this distribution are. So now you don't actually have to sample this whole distribution to figure out where the main modes are and where the tails are of this, but you can just immediately take the network output, this uncertainty score, to have an idea which parts are physically good in distribution and which ones aren't. And again, I think this has a lot of practical use cases where you can really flag bad samples based on this uncertainty score. Okay, the last thing I wanted to show in the scope of this paper is the fourth point in my recipe that I mentioned at the beginning of the presentation, where once you have a good distribution learner, you also want to be able to extract the most relevant samples for your use case. And the approach that we pursued in this paper is a fine-tuning approach with preference alignment. So this is inspired by the DPO algorithm that is commonly used for fine-tuning large language models as well. And the idea is rather simple. So we take our reference model, DRUGFLOW in this case, and we sample a bunch of molecules. And then we assign to each sample a user preference. So this could be anything really. So it could really just be a human labeling which one is good, which one isn't good. For the sake of our benchmarks, we just use the computational score or different computational scores, defined a certain threshold, and then said, okay, everything that's better than the threshold is a winning sample. Everything that's worse than the threshold is a losing sample. And then we train this a new preference aligned model that initially starts from the same weights as our base model, but is trained with the loss function that increases the likelihood of our winning samples while decreasing the likelihoods of losing samples. And it also has some regularization term that forces it to stay close to the reference model, because you don't want to completely wander off and like get into this trap of, let's say, mode collapse by generating the same sample that just happens to score well all the time. And yeah, I promised some benchmarks. We did this with 4 different metrics. RIOS is a collection of structural alert filters. So basically, if you see a certain substructure in your molecules that is flagged by these RIOS filters, you don't want it for different reasons, and then they're bad. Then we have again this drug likeness score that I already mentioned a couple of times. Same for the docking binding affinity score and synthetic accessibility. And then we have 5 different bars in each of these experiments. The first bar is the training set, how examples from the training set score in these metrics. The green second bar is the reference model, the baseline drug flow model. And here's already the first nice observation is these values are usually very close to the training set, again, speaking to the distribution learning capabilities of drug flow. Then as another baseline, we have a simple fine-tuned model. And by that, I mean, we just take the winning examples, completely ignore the losing ones, and fine-tune our drug flow model just based on the winning example. So basically, yeah, the simplest fine-tuning strategy you can come up with. And this also works to some extent. You see that all the metrics improve at least a little bit doing this. But it doesn't get close to the preference alignment, which really seems to be more efficient at doing all of this. So here we have two different models with the preference alignment scheme in each case. The one with the solid bar is kind of the vanilla preference alignment on one metric. But these like dashed bars, it's a preference aligned model that optimizes all four metrics at the same time, not just a single one. So it's actually the most powerful model. And even this one performs quite well, even as a general model. Yeah, this is just to show that we can take a strong distribution learner and then shift the distribution that it learns towards more favorable spaces for our use cases. Okay, that's already the last piece of data I wanted to show. Some main takeaways that I see from this work. Well, the first point is actually more about the motivation part. I think generative AI really has a huge potential to play a role in early stage drug discovery and just make it more efficient in the future than it is right now, and kind of circumvent this E-rooms law that I showed, and hopefully make new medications much cheaper in the future. But I hope I could also convince you that if you train distribution learning models, like these generative models, classical diffusion models, large language models, and so on, you should really also focus on benchmarking them correctly according to their objective. Because then you can really focus on making the best distribution learning model without worrying too much about any absolute metrics that you care about downstream. And instead, that's the last takeaway, you should do this at inference time when you already have your powerful model. And there are different techniques. There's the fine tuning, but there are also some sampling techniques that I think I will not get into for the sake of time. But there are some other things that you can do to really optimize molecular properties and have some like more target specific improvements. Maybe I want to conclude still with this Outlook slide. In theory, I prepared slides about each of these points, and I can go a bit more into detail of them. But yeah, for the sake of time, I will just keep it at this slide. And if you have questions, I'm also happy to show you the slides later on. So where I think this work should be going in the future to make it even more practically relevant is we should work on the training data set. Because yeah, I already mentioned before that this is not something we looked into in this paper, but it is of crucial importance for generative models. We can optimize molecules while we're sampling them. It doesn't have to be a training scheme. This is unfortunately something I couldn't really get into. And this is nice because then we can also impose some synthesis constraints, meaning that we can generate molecules that we can easily make. And that also completely unlocks experimental validation, which has kind of been an issue in this whole drug discovery, ML for drug discovery field, that most people just never tested the things they developed in the wet lab just because they couldn't make the molecules and hence also couldn't test them. And this is something we're very actively doing right now. We just want some real world evidence that the things we're developing are working. Okay, but with that, that's basically what I wanted to show. And again, if you have some questions about these points, I have some backup slides that I could show to answer them as well.
Okay, thank you. Thank you, Arne. Thank you very much for this wonderful discussion. I love the closing remarks, and I think I'll start from there. For someone who is new into drug discovery, what is that one sentence you would say for to be like the goal of drug flow? Yeah.
I would say the goal of drug flow was really more we wanted to approach the problem properly. Basically, how I motivated in the beginning with these slides that people, in our opinion, just didn't benchmark the models in the right way for a long time, and we did it ourselves also a bit incorrectly, because that kind of prevents progress on the ML side of things. because then all of a sudden your new technique that you came up with doesn't actually score better because people are not measuring the right thing, basically, and then it's hard to compete, even if you have like good ideas. So this is kind of what we wanted to focus on in this DRUGFLOW work, and what DRUGFLOW does. I think it's also still one of the better models out there already for generating small molecules, but it is still missing some of these crucial components that I mentioned on the Outlook slide. It's not by construction that the molecules are synthesizable. So you might have some designs that look nice in the computer, but you will never be able to make and test them. It also isn't trained on a huge data set. So the chemical variability that you get in your outputs is rather limited. So this is really, I would say these are core features that you should have if you're trying to do this in practice and you want to use these generative models for real world applications. And this is also precisely why we're working on it now and making that our main focus rather than, let's say, innovating on the machine learning side more.
Yeah, one thing when you introduced it, I think it was figure 1, sort of like the overview of DRUGFLOW.
Yeah, I can go back.
My interest is, or my intuition is about the architecture. And I think looking at the paper looks to be somewhere a GNN, but overall, inside the architecture of DRUGFLOW, anything you can say about it?
Yes, I think I also have a backup slide. I try to stay purposely a little bit vague on these details because I didn't want to get stuck in things that are not actually core contributions of the work. And I think the network architecture is rather straightforward and not necessarily a contribution of this work. But you're right, it's a GNN. I guess the one thing we did a bit differently than at least us ourselves did in the past is we didn't encode the protein and the ligand in the same space, basically have only one node type with like, let's say a flag that tells the model which is which, but we keep them separate throughout the network. So there's like some nodes and some trainable parameters dedicated to molecule nodes, some dedicated to protein nodes, and they have all their own sets of parameters and they can be optimized independently. And the nice effect this had on in practice for us was that we could scale the model to a much larger network size. Our previous model had 3 million parameters roughly. This one has 12 million parameters, and it's mainly because with the same number of computations, we now have more trainable parameters because we're keeping these like separate tracks for the protein and the ligand.
Okay, so does that mean like when you look at the first layer of the architecture, the ligand, the protein, the ligand edges and protein edges, and then the interactions, all these are encoded and they are inputs into the GNA?
Yes, exactly. So the inputs are things like the atom type, or the protein residue type, or the bond type, you know, if it's a single, double, or triple bond. So this is a feature that lives on the bond, for example. And all this is input to the network. Yeah, I think that's pretty much it, actually, what goes into the network.
Okay. Wow, sounds pretty impressive. Like I'm wondering if you this sort of encoding and representation, how it is done, but I think that could be a bigger sort of explanation.
Yeah, I mean, in the end, it's all rather basic, I gotta say. We also didn't research this too much or compare too many different things. The categorical features like the atom types are one-hot encoded, like you would probably do as a first try anyway with any categorical variable. What I didn't mention is this is an equivariant architecture. I'm not sure if this is necessary, but basically for historical reasons, we stuck to it. Which means we encode scalar features and vector valued features separately as well, making sure that vector valued features are transformed in an equivariant manner. If you transform your input, your output will transform equivalently. And I mean, this, for example, a vector valued feature would be the difference vector between two atoms. That's encoded really as a vector in 3D and then processed by the network as such.
Okay. I think the last for me is about the Markov bridges somewhere.
Yeah.
You talked about the Markov bridges, and I think you used them for sort of like encoding the different types, like a bond types and
Exactly. So this Markov bridge model is something we developed for a previous work where we tried to match products to reactants. Let's say if you want to predict how to make a molecule, if that's your task, you need to map products to the reactants, the precursor molecules. And it's basically just an extension of a discrete diffusion framework that already existed before. Because usually if you have discrete diffusion, you start from some categorical distribution, let's say a uniform categorical distribution, and you go to some like some distribution that has all the probability mass on a single category. Here what we do is we start from one category and transfer all the probability mass to a different category in this Markov bridge model, which kind of just allows us to have arbitrary priors in a sense. So this is what we do. Basically, we sample a category in the model. When we create a sample from the prior, we sample one category, and then the model converts that into the final output category, which, for example, is the atom type.
okay. I think that's all from me. I don't know whether the members in the audience will have questions. Members, if you have any questions, you unmute yourself. Otherwise, if there are no questions, I want to thank Arne for the wonderful presentation. Thank you very much. This has been great, and it's a great introduction into distribution learning also. And
Yeah,
anyone,
thank
I
you
would
very much again for the invitation. Thanks you all for being here.
yeah. So I invite you members to maybe read the paper. One last question, I think I had forgotten. Is there access to code?
Yes, the code is on GitHub. I think you should find the link in the paper as well.
Okay.